It's a real honor to have Dr. Dick Atkinson give us a kickoff welcome this afternoon. Dr. Atkinson, if you aren't familiar, was for 5 years the president of the University of California system. Now, if you don't know what that is, that's like the whole system. So in my neighborhood in Brooklyn, we would call him Capo di Capi. But more importantly, before that, he was for 15 years the chancellor here at UCSD. During the time that UCSD built its reputation as an international renowned university for research and training. So we really owe him that gratitude of thanks. And without further ado, I give you Dr. Atkinson.
Thanks very much for that introduction. I wish it were the case that I think I wish it were the case that I was president for only 5 years. It was much longer than that. But it's a pleasure to be able to welcome all of you here to the UCSD campus for this symposium honoring Palmer Taylor. In my remarks today, I'm not going to review Palmer Taylor's long list of honors and awards, although I can't resist mentioning the Legion of Honor by the President of France last year. I'm also not going to comment on his remarkable scientific career and his mentorship of so many young scientists and graduate students. Rather, I'm going to comment on his role as a faculty visionary, a faculty leader who played a critical role in the evolution of the medical sciences at UC San Diego, and particularly his vision and plans for creating a school of pharmacy and pharmaceutical science. I'll conclude my remarks today by stating that Palmer Taylor was key to the development of this school. If there had not been a Palmer Taylor on the faculty at UC San Diego, there would not be a School of Pharmacy here today. Palmer joined the faculty here in 1971 as an assistant professor. There's some great pictures of him that have been across there as a young man. To tell you a little about what the School of Medicine and what the university was like in those days, I just want to track back a little to the origins of this particular campus of the University of California. The campus really dates back to the turn of the last century, 1900, when we established the Scripps Institution of Oceanography. It was a small research station that prospered. It was excellent research, but a very small operation. But in the 1950s, the regents of the University of California made the decision to establish a general campus of the University of California here in La Jolla. And the faculty that were already in place at the Scripps Institution of Oceanography had responsibility for recruiting the initial faculty and for laying out programs for a future full-size campus of the University of California. The members of the Scripps faculty, they were principally interested in oceanography and geophysics, and they were a great collection of brilliant physical scientists. And when it came to establishing university departments in mathematics, biology, physics, and chemistry, they understood, or I should say mathematics, physics, and chemistry, they understood the disciplines and recruited outstanding individuals. In those early days, we recruited a number of people that were already members of the National Academy of Sciences, and there were several Nobel laureates. However, when it came to the issue of the biological and medical sciences, the Scripps faculty, being principally physical scientists, were not, they were on less certain ground as to how to proceed. And after much discussion and debate and consultation with leading biologists and biological scientists around the world, they made the decision to focus the new biological and medical programs in a somewhat non-conventional way. They were going to focus them principally on the emerging fields of molecular and cellular biology. At the time, that approach was not in vogue throughout the, remember this is late 1950s, early 60s. That approach was not in vogue at that time in the academic world. Nevertheless, the faculty were convinced that the future was in that direction, and of course, time has proven that they were correct. Well, that's the situation that Palmer Taylor walked into in 1971 as an assistant professor, very junior faculty member, but he quickly rose through the ranks, and by 1978, he was a full professor, head of the Division of Pharmacy, which was part of the Department of Medicine, which was part of the School of Medicine. I came here in 1980 as the new chancellor. I knew about Palmer Taylor's research on acetylcholine receptors, and I looked forward to meeting him. When we first got together, we quickly passed into a discussion on the future of this institution, in particular his view that there was a great need for a school of pharmacy, and a school of pharmacy that had a strong focus on pharmaceutical sciences and research. It didn't take Palmer long to tell me about this vision for the school, and throughout the 1980s, Palmer made repeated efforts to lay out plans for such a school, and he enlisted strong faculty support, and he and I on many occasions went up to the president's office, met with regents of the University of California to discuss the idea of a school of pharmacy here at UC San Diego. But we had very little success. The answer was always that the University of California already had a school of pharmacy, a second wasn't needed, and if the San Diego campus felt that they wanted to be involved in the training of pharmaceutical scientists and students, they should work out some type of joint arrangement with UC San Francisco. Well, we did that, but it wasn't what we were looking for. It was not what Palmer Taylor had envisioned, and so we struggled through the 1980s. But things began to turn rather dramatically by 1990, the early 1990s. And there were three factors in that turn. One is that by then, the medical school and the biological sciences had already been established as an outstanding program here at UC San Diego. The second factor was that the biotech industry was on the move in San Diego. There was a lot of activity in biotechnology, and UC San Diego was clearly the catalyst for much of that activity. But a third factor was rather surprising and I think quite critical, and that was the fact that in 1994, the National Research Council, part of the National Academies of Science and in Medicine and Engineering, the National Research Council of the United States issued a report on the quality of graduate education in the United States. And what they did was they studied 41 different programs, doctoral training programs in the United States, and it was principally a reputational survey, but I think quite well done and quite well received. And what they found was, what they did was rank these 41 programs, doctoral programs throughout the country in terms of the quality of the graduate programs, the quality of the graduate faculty, the quality of the products from the programs. And what was so surprising was that pharmaceutical sciences at UC San Diego was ranked 3 in the country. That was quite surprising. There was no school of pharmacy here, but number 3 in the country. That study was quite significant for UC San Diego as an institution, because when you look at the study, it's just simply ranking each individual area. That is, each individual area, for example, physics, who's 1, 2, 3 in universities and so forth. But quickly, a group got together that looked through the report and composed their own version of the study, which was to average through in a very appropriate way to get a ranking of universities in terms of the quality of graduate education. And I just happened to bring that book along. It's interesting, as I said, pharmaceutical sciences ranked number 3 in the country, but for the universities as a whole, the ranking was as follows. There were the top 10 universities, there were 2 publics and 8 privates. And the ranking that came out in this change study was MIT 1, Berkeley 2, Harvard 3, Caltech 4, Princeton 5, Stanford 6, Chicago 7, Yale 8, Cornell 9, and UC San Diego 10. That was quite stunning. Remember, this is 1994. It's a long time ago. And the authors of that report go on to say the following. They talk about the University of California system being rated extraordinarily well. Then they talk about UC Berkeley and how exceptional it was. But then they go on to say something about UC San Diego, and I'd like to just quote it. UC San Diego rated extraordinarily well, particularly for institutions that became a campus as recently as 1964. It was rated 10th in mean score, and so on and so on. And then they point out that it ranked much higher than older and larger UCLA, higher than any public university campus in the United States except for Berkeley, and higher than such highly regarded private universities as Columbia, the University of Pennsylvania, and Northwestern. Once that report came out, once all of the attention was focused on biotechnology and UC in the San Diego area, there was a new enthusiasm to examine the notion of a school of pharmacy here at UC San Diego. It turned out that the governor at the time, Gray Davis, was very focused on the idea of technology transfer from research into application. He visited frequently. He met with scientists here in the pharmaceutical sciences and in other areas of the university. A number of regents became very excited by what was going on in San Diego. And once again, Palmer-Taylor was asked to prepare a report to establish the School of Pharmacy here at UC San Diego. Well, it was not a tough job for him. He'd done it several times before, but that report was prepared, and it was received by the regents in the year 2000 when they officially established the SCAG School of Pharmacy and Pharmaceutical Sciences. As I noted at the start of my remarks, I can't read too well anymore. I can't seem to see what my notes say here, but as I noted at the start of my remarks, we would not be here today if it were not for Palmer-Taylor. From the very beginning, it was his vision for the school, his determination, his effectiveness in making the case that made it possible for the school. So I conclude my remarks by saying that I want to thank him for the University of California and for the UC San Diego campus for the brilliant job that he's done, both as a scientist, but in particular in his helping to formulate and establish the school. In fact, standing in this room, my thought is that something should be named in his honor, given his remarkable contributions. And I think what should be named in his honor possibly is this very auditorium. Something I'm going to think about. Thank you, Taylor.
So we'll go ahead and just get started with our first speaker of the afternoon, and that's Dr. Gabi Amitai. He is from the Wall Drug Research Institute, the Weizmann Institute of Science, and he's going to be speaking on structural and functional studies on the reversible and irreversible inhibition of acetylcholinesterases.
Good afternoon. I really feel honored to present here at the conference honoring Palmer Taylor. What you see here on the screen is a picture taken by a friend of us, Alvi Berman, in '79 when I first visited the UCSD looking for a place for postdoc when I almost finished my PhD at the Weizmann. I started my career actually and worked for 45 years in the Israel Institute for Biological Research, which is the National Center of Israel for Chem and Biodefense Research. And just recently, in June, I retired. So I wrote Palmer a short note that I'm going to retire in June. And his response was that is my first postdoc that retires before me. So what you see here in the picture, you recognize Palmer, but this is the guy who stands near Palmer is me at the age of 31, and Palmer is 40 years old here, which struck me. He's younger here as my younger son today. So this is like a time machine, something that we have to be accustomed to. Palmer is a real, I consider him as a real teacher, and one of his mottos was, when he came back from Cambridge, he did his sabbatical in Cambridge. He told us, all the postdocs in the lab, that in order to do good science, you have to have two basic things. One, you have to ask a good question. And the second one, you have to find a good experimental system to answer that question. One without the other is worthless. And it's true. It's a motto that really follows through all our career, all the postdocs that worked with Palmer. And I think Palmer is a mentor, as was mentioned previously, an excellent scientist, and I consider him also personally as a mensch. And if you don't know what's a mensch, go to Google and see the translation. Okay, so the topic of today is actually, I will try to present to you our efforts to develop new small molecule scavengers. People are trying to develop large molecule scavenger, catalytic, stoichiometric, but we try to do something else, which is, I'll tell you in a moment. And the work was done at the Israel Institute for Biological Research, the Department of Pharmacology, and it was supported by the US DoD, DITRA, for the last 3 years, just before I retired. So let's look at the bad guys. What we see here is a collection of molecules that the common denominator is here in the center of each molecule, which is the phosphorus. And the phosphorus is a tetrahedral phosphorus, and all the nerve agents are asymmetric phosphorus organophosphates, organophosphate compounds. And it means that there are at least two optical isomers, except for this case, someone, where we have two couples of two diastereoisomers, and the minus isomer is the more toxic one. So that constitutes some kind of a toxicological problem, but this is the story about these bad guys, that all of them are inhibiting acetylcholinesterase covalently. And this guy, Soman, after inhibiting the enzyme, dealkylates and forms the resistant non-reactivatable conjugate, which is called aged acetylcholinesterase. So not only the nerve agents are very toxic, but there are some pesticides, insecticides, like this guy here, monoprotophos, And you probably know about this very sad case in Bihar, India, in 2013, and this is the location of this small town. So medical help didn't get too quick into this area, and there were 23 to 27 casualties, death cases, and other 25 casualties of poisoning, severe poisoning out of a food that was poisoned in this case in a school. So school children got poisoned from the oil that was mixed with the organophosphate. Now, from the WHO reports, there are 250,000 death cases per year out of OP pesticides, and 30% of the suicide cases were done with OP pesticides. So it's a real severe toxicological problem. And to remind you that the nerve agents, organophosphates, sarin, soman, and cyclosarin and VX are really toxic. These are the doses per body for 70 kilos. So what you see here, that the most volatile nerve agent, which is sarin, that was used in the Tokyo subway case, the terror that was done by underground movement, and in the civil war in Syria during the last 4 years, there were cases of using this sarin. It's the more toxic, the more volatile compound. It's more toxic through the inhalation and less toxic through skin. But there is an exception here. The VX is both toxic. The lower the dose, the higher the toxicity, both in the inhalation and the skin. Just let's imagine that 10 milligram per person is less than a standard size of a drop. So what happens in the cholinergic synapse? When acetylcholinesterase is inhibited, acetylcholine that is released from the presynaptic membrane accumulates and creates an excessive amount of acetylcholine, and we need to reactivate the enzyme on one front here, and also block the receptor with atropine and other anticholinergics as a second front. So we're using reactivators like oximes to reactivate the enzyme. If we know that the people are going to enter a contaminated area, we use pyridostigmine as pretreatment to partially block the enzyme. And also in severe cases, we add to the mixture some GABA agonists, such as the anticonvulsants, such as diazepam and midazolam. So these compounds will save life, they will accelerate recovery and return to full capacity, and they prevent delayed and long-lasting neurotoxicity. And here are some samples or examples of the oxime developed starting in the 50s in Columbia University by Ernie Wilson and Sarah Ginsberg, by just using their intuition, because nothing was really known about the 3D structure of the enzyme. What they knew that there is an esteric site and an ionic site, a negatively charged ionic site near the catalytic site. So they designed a molecule, which is a pyridinium, metal pyridinium, and in position 2 or position 4, they put a soft nucleophile, which is the oxime, the hydroxyl, the aldoxine group here, or in 4 position. In 3 position doesn't work for some reason. And then later on, in the late 50s, there were like bis oxime developed, like TMB4. And in the late 60s in Germany, in Hagedorn lab, there was this HI6 bis quaternary oxime developed, which is considered one of the most potent reactivators known up to date. Palmer and his group, together with Dr. Sharpless, developed a new generation of oximes that are non-quaternary. So there is no real quaternary nitrogen in this molecule. And by developing these non-quaternary, they got the blood-brain barrier permeability. There is orally bioavailable to maintain the plasma and tissue levels and all other advantages and traits that you see here. And it was published by Palmer and Dr. Sharpless just recently in 2012. And what we know about these antidotes, that they are symptomatic. So they cannot really detoxify the toxins, the organophosphorus toxins. And people have tried to use various approaches in order to detoxify the organophosphate by either using enzymes that are stoichiometric, like cholinesterase. It was a big project funded by DITRA to develop acetylcholinesterase as a stoichiometric and then butyrylcholinesterase. And then they developed the catalytic bioscavengers, the OP hydrolases, PON pyroxonase, and the FPAs. And recently, Palmer-Taylor, together with the lab of Barry Sharpless, developed this clever approach of having an oxime-assisted catalytic detoxification of OPs by cholinesterase. They combine an oxime with a stoichiometric scavenger, and by that they transform it into catalytic bioscavenger. What I try to show you today is another approach, which I call a small molecule scavenger. And the idea is to combine two different moieties in one molecule. One moiety will be a nucleophile that detoxifies directly the OP, will react with the organophosphate in solution. And the other moiety will be a reactivator. And the soft nucleophile we used for detoxifying is hydroxonate. And for reactivation, we of course took the oxime because it was a successful reactivator. And just to show you the concept of a hybrid, bifunctional hybrid, as shown here, you see there are scissors here that will cut the molecule, and there is a small spacer that binds to another part of the molecule which looks like a fishing rod. And if I operate this simple animation, what you see is that the scissor will cut the leaving group from the organophosphates that will detoxify the organophosphate to form the phosphonic acid. And then if it evades the scissor, then it will go into the active site and inhibit. Then there will be a small fisherman here sitting here on the rim of the gorge, and it will take it out. So be patient, and that's it. That's the other. So that was the concept. And by using this concept, we thought that we'll have the following advantages. Of course, if you have a small molecule compared to a large molecule that is like 40 to 80 Ka Dalton, and the small molecule is a 200 to 400 Daltons, then you have more moles per gram. It's between 80 to 164, depending on what you compare, if you compare the pyroxonase or the butyrylcholinesterase. Secondly, by combining both detoxification and reactivation in one molecule, we will have unitary pharmacokinetics for both activities. Thirdly, pharmaceutical application is easier if it's soluble, and we chose such moieties that will be soluble to start with. The compatibility with currently used small molecules is another advantage, and of course, if we follow what Palmer and Palmer-Taylor and Dr. Sharpless did, we thought that we also can use the cholinesterase in blood as a kind of landing dock, temporary, that will be inhibited and then will be reactivated, so we'll get some kind of a turnover effect. So if now we translate this simple animation into chemistry, we see here that we took a pyridine ring with hydroxonate as our cutting moiety, the scissors. And on the other side, we took and linked to it pyridinium aldoxime, which is a 2-PAM. This is the antidote which is still in the syringe of every soldier in the US Army. And the linker should not be too long, so it will be either methylene or propylene or other short spacer that will bind these two moieties. Now, interestingly, if you look on the hydroxamate as a nucleophile, one can ask, well, you can use pyridinium hydroxamide as a reactivator, and not only as a detoxifier. It turns out that the hydroxamates are very, very poor reactivators of OP-inhibited cholinesterases. For example, it's like 50,000-fold less efficient as a reactivator of serine-inhibited enzyme than the 2-PAM. On the other hand, this nucleophile, the aldoxine, is a very poor detoxifier, but is a very good reactivator. I don't think that anybody knows what it happens, but probably maybe the oxygen here makes it a little bit too tight in the active side to be a good reactivator, where this guy is more compact in its size. So we synthesized 22 hybrids and selected 3 out of them that showed us the best profile in vitro. It's the 2-PAM with a spacer for propyl here and 4-pyridine hydroxamic acid, 2-PAM with a metal linker here, and benzyl hydroxamic acid as the nucleophile that will interact with the organophosphate directly. And the third one is we're having here 2 oxime groups on the same pyridine, as in one of the oxime that was developed in Germany, the HLO7, and on the other side of the molecule, benzyl hydroxamic acid with a metal spacer. So in the lab, we gave it the short name, the propyl, the methyl, and the DPAM, just not to repeat these long names. So the first experiments we did, we wanted to know if these hybrids are really detoxifying the organophosphates. And indeed, we measured the kinetics of decomposition or degradation of VX, soman, serine, and cycloserine by a number of building blocks and our hybrids, and we got the kinetics summarized in this table. And what we see here, that the 3 hybrids that we chose for further studies are here: the methyl, the dipam, 2,4-dipam, and the propyl have a larger rate constant than 2-PAM in hydrolyzing serine, cycloserine, and so on. With VX, we couldn't hydrolyze it, probably because the PS bond is too resistant for a nucleophile to decompose it. So these are the numbers that I emphasize here for serine and cycloserine. With reactivation, we tested all the hybrids, and they were comparable to 2-PAM. So we had the feeling that we didn't lose the reactivation potency by combining the 2-PAM moiety to an hydroxamate. And for example, this propyl, the propyl hybrid here, with this K observed, when we ran the bimolecular analysis, we got a 30-fold more potent reactivation activity for the propyl when compared to 2-PAM. So we asked ourselves why the propyl is so active compared to the other guys, and we knew that the lab in the U.S. Army, who is working also on developing antidotes, chose this compound as a candidate to replace 2-PAM. They call it ICD, the Institute for Chemical Defense 585. And the only difference between this molecule and our hybrid is in this part. Instead of a hydrogen, a proton here, we have an OH. So it's very similar in the structure, and both are very similar in the structure to HI6, which is the most potent reactivator. The only difference is here the oxygen is a linker instead of the methylene. So the structural resemblance to active reactivators probably explains why this compound is very active. When we compare the reactivation potency of this propyl hybrid to other known oximes, it's not in the Premier League. It's probably, it's shown here that it's weaker as a reactivator than the known oximes. But still, it's in terms of practical terms, in toxicologist, in vitro toxicology, it shows enough activity to reactivate at least part of the enzyme which was inhibited by serine, cycloserine, or VX. Now in the course of studies, we asked ourselves whether these compounds could protect, temporarily at least, the active site from inhibition by organophosphate. And the idea was that this quaternary nitrogen of the pyridinium ring will interact with the anionic site of the active site of the enzyme, and at least temporarily will delay the inhibition by this guy, the serine or any organophosphates, which eventually will inhibit it irreversibly. So what we did in order to measure the affinity of our compounds to the enzyme, we measured the concentration dependence of the reversible inhibition of acetylcholinesterase by each and every hybrid that we synthesized, and also compared it to known oximes. So what strikes the eye immediately that our compounds are have higher affinity than most of the oximes, except for HI6 and HLO7. So, and when we looked at the IC50 values, we saw that the three hybrids, the methyl, the propyl, and the dipam, and also HI6, are in the range of 10 to 50 micromolar. When one is trying to calculate what's the concentration of an oxime injected in vivo as a therapeutic agent, the concentration in blood, the average, I would say, concentration will be in this range. So it's really relevant to have these concentration in blood during the treatment. So maybe these compounds protect the enzyme from the irreversible inhibition. So indeed, we measured the kinetics of inhibition by various organophosphates in the presence of some of our hybrids. And what we see here is the time course of inhibition without any protection, say for the this is dipam here, dipam. And the higher the concentration of the hybrid, the slower the kinetics of inhibition. This in this case, and also in the methyl case, less with 2-PAM and the third hybrid. So what we get here is slowing down of the irreversible inhibition of acetylcholinesterase by the organophosphate. And we gave it a quantitative measure by plotting the 1 over the K observed versus the reciprocal of the concentration, and we got a slope that really gave us a sense of quantitative measure of the protection of the enzyme from irreversible inhibition by the covalent ligands, by the nerve agents. And the most active ones are the methyl and the dipam here. The propyl probably is less protecting the enzyme, but because it's a good reactivator, it compensates for total activity. The in vivo studies, first we measured the toxicity of these compounds. Now, to start with, we chose 2-PAM, which is relatively non-toxic drug, and the benzhydroxamic acid and the 4-pyridine hydroxamic acid are known as relatively non-toxic, and indeed the toxicity of the compounds are, for this guy, for this hybrid in rats, it's about 200 milligrams per kilogram. In guinea pigs, it's a little bit more toxic. The methyl is 180, 113, and 2,4-DPAM is above 200 in both species. Just to show you that we also tested some other hybrids with another kind of a bridge between the two moieties. This compound was a good detoxifier, but it was also relatively toxic, the 13 and 6 milligrams per kilo, so we didn't pursue with this compound. And this is the toxicity of 2-PAM as a reference. The antidotal efficacy that we measured, and this is going to be the last 3 slides of my presentation, we tested the antidotal efficacy in 2 modes. One mode is a pre-treatment. We first inject the animals with the mixture of one of our hybrids, or 2-PAM as a reference, in the presence or in conjunction with atropine, because you need atropine for getting any antidotal efficacy. The other mode is what we call post-treatment. We first expose the animals to the agent, and then 1 minute later we inject the mixture of one of our hybrid and atropine. And the results are shown here in rats. The red bars are post-treatment, and the black bars are pre-treatment. And the numbers here designate the protection ratio of each and every compound that was tested. 2-PAM, and here's the, I see the methyl here, the propyl, and the dipam. And what we see here, that the methyl and the propyl have significantly higher protection ratios than 2-PAM that we use as a reference against sarin poisoning. Also against cyclosarin, the propyl stands out with a relatively high protection ratio. With VX, all of them, most of them are very active. And with Soman, nothing happened, or nothing was active as a pre-treatment, only as a post-treatment, when we use pyridostigmine as a pre-treatment, as was used by many other laboratories. In guinea pigs, again, we get a similar picture where the pre-treatment, the black bars, gave us higher protection ratios as compared to post-treatment. And in the post-treatment, our hybrids were as active as 2-PAM. We got it against sarin, the cyclosarin. In the Soman case, it also worked as a pre-treatment without pyridostigmine, which is remarkable. And with VX, we got similar results, a little bit lower than the 2-PAM, the standard treatment that's used by the US Army as the antidotal mixture. So if we sum up, we synthesized, as I told you, 22 compounds, and we selected only 3. We show that there is a detoxification effect and reactivation effect caused by the same molecule. These hybrids were detoxifying and reactivating OP-inhibited enzyme. The antiphosphorylation activity, as I showed you, was really functional. It inhibited reversibly, but also retarded the inhibition by the nerve agent. The in vivo toxicity, the acute toxicity of our hybrids is they are relatively non-toxic in both rats and guinea pigs. I didn't have time to show you that we did some in vivo reactivation in rats and guinea pigs. We showed it by using sublethal doses of sarin, and these compounds showed a very high reactivation or regeneration of whole blood cholinesterase in these sublethal exposure to sublethal doses of sarin. And this is the in vivo results that I just mentioned. And what we selected as our MVP is this propyl, which is both a good reactivator and very efficient detoxifier hybrid. And last but not least, my friends in the lab at IIBR, Nissan Ashkenazi and Nira Barner, who did the organic synthesis, Reli Gez, Shira Chapman, Lily Rove, Eti Grauer, who did all the work, the in vitro and vivo. And this was supported again by the US DoD, DTRA, between 2012 and 2014. And I really thank you for your attention.
I think we have time for a couple of questions. Over here.
Very nice talk, Gabby. Have you tried the efficacy, or maybe it skipped my attention, against pesticide, OP pesticides?
Oh, no, we haven't done it. Well, I wish we could do it, but we were funded to do the nerve agents. That was the interest of the agency.
Second question, any idea about butyryl interaction?
Butyrylcholinesterase?
Yes.
Oh, yeah, we did some work. We did some reactivation studies. Some of the compounds reactivated butyrylcholinesterase that was inhibited by some of the organophosphates, but not all of them. I don't have the data here, but if you're interested, I can send it to you.
Because amount of butyryl in blood is about 10 times more than acetyl.
Right, right. But you know that there is like 80 nanomolars butyryl and 3 nanomolar acetyl in blood. But in terms of activity, they are 1-to-1, because acetylcholinesterase is much more active as an esterase as compared to butyryl.
But here you would, if you are looking at detoxification, then you would be looking.
I know what. Yeah. So you get the turnover effect by reactivating the butyryl more than the acetyl.
Then amount of protein actually, or
molarity will play. Yes. Let's see what's.
Well, I think we'll move along. Let's thank our speaker once again. It's really a pleasure to introduce my colleague and good friend, Bradley Moore, who shares a joint appointment between the Scripps Institution of Oceanography and the Skaggs School of Pharmacy, and has been here about 10 years now as well. And I think it's fair to say that his program has just blossomed under the cultivation of what UCSD has to offer in terms of the environment and the culture, much of which Palmer has helped to create. So Brad is speaking on the rapid and evolving world of natural product science and its impact on human health.
All right. Well, thank you, Bill. You know, it's just a delight to be here and to be part of this symposium honoring Palmer, and who's really the reason that I'm here, and Bill and a number of other colleagues who work with natural products here in the School of Pharmacy. And it's just a real pleasure to be able to share some of our work with colleagues and friends who are here. So I won't belabor the point that small molecules from nature have really impacted human health in profound ways. And this field was obviously given greatest recognition just a few months ago with the Nobel Prize in Medicine for discoveries of a number of infectious disease agents that impact hundreds of millions of the world's poorest people on this earth. And our very own new dean, Jim McCarroll, wrote an article for this journal, Natural Product Reports, detailing some of the work by these investigators because of his interest and expertise in infectious disease and work as well. Small molecules in nature have not just impacted humans, but also are used routinely by many of us in the field of pharmaceutical sciences as tools to explore how cells work. And many of the earliest proteins, before they even were discovered or had names, were often given their names based on the natural product molecule that they interacted with, whether it be a bacterium and how it made cell walls interacting with penicillin. We call this penicillin binding protein. Before we had a name for it as transpeptidase, or mTOR. I had a class earlier this morning on hypertension with the pharmacy students, and we were talking about the ryanodine receptor and its role in calcium uptake. So that goes on and on. We see this, and I think Palmer recognized the importance of small molecules from nature in the role of pharmaceutical sciences quite a few years in his career. And before the school was formed, he interacted with scientists down at Scripps. One, the very noted marine natural product scientist, John Faulkner, and the two of them published papers together, including this one here on a small molecule from a shellless mollusk that would produce this very toxic molecule that was interactive with acetylcholinesterase. So at that time, I think the recognition of the partnership between the natural product chemistry emphasis here at UC San Diego, and most of that emphasis was down at the beach at Scripps Oceanography. And when the new school was being started here, there was the realization that I think Palmer had the opportunity to really unite what was something unique about this campus, the only campus in the United States, which has a school of pharmacy and a major oceanographic institution. And that's where a number of the faculty appointments were, being able to bridge those two departments. And what I think is remarkable about many of the molecules that I shared with you earlier in this presentation is it's remarkable to think that today the science of natural product discovery is fundamentally more or less the same as it's been for the past almost 100 years. We look for a molecule and we look at it either using a chemical means or analytical technique, or we look for some kind of biological screen and we find a molecule. But for the most part, when you do this kind of work, it's been called grind and find chemistry because we're using sort of what nature provides as our starting point. And what I'd like to share with you today is I think there's a new way of doing this that takes the advantage of genomic information that's pretty much an orthogonal sort of thought process of how we can discover chemistry from nature and what it's doing and its roles in nature as well. And I think really the ability to rapidly sequence genomes is really teaching us fundamental lessons about the chemical language that's out there between cells. And being able to understand sequence, we can combine this information and think about the small molecule chemistry that a cell may harbor. And this is available to us because of obviously the rapid increases in sequence technology that makes it where any one of us in this room can sequence the genome of an organism fairly affordable and rapidly to do so. And I think also the realization in the microbial world that genes that encode enzymes that make a specialized molecule are typically clustered together in the genome. Together with not just the enzymes that make the small molecule, but everything else that goes along with it, whether it be the regulation of that pathway, when to turn those genes on, when to turn them off, maybe an immunity factor if that molecule is toxic to the cell, maybe a transport mechanism. You get this remarkable package of information. In many organisms, I think when the first genomes were coming out, I think the community was quite astounded to find that an organism didn't just produce maybe one natural product that made whether an antimicrobial agent or an anti-cancer agent, but not two, sometimes 10, 20, 30, 40, 50 molecules, maybe 10%, 15% of the entire genome would be dedicated to making a smorgasbord of small molecules. But if you take that organism into the laboratory and you grow it up, you find a small percentage of that chemistry. So I think the field has been realizing there's quite a bit going on here, and most of it is hidden to us. So let me tell you about what a gene cluster is to introduce this terminology to you. And if you imagine a small molecule like the one here, a small molecule is built from building blocks. Those building blocks are stitched together by a number of enzymes to make intermediates. Those intermediates then turn into a molecule. So we can understand the enzymes that do this. We can sort of cluster those, think about those enzymes as a little package. Each of those enzymes is encoded by a gene. Typically those genes are co-clustered together in the chromosome. And therefore there's a one-to-one correlation between a set of genes and a small molecule. And you can go in two directions. You can sort of think about what the structure of the small molecule is and how you can connect that back to genes based on biochemistry or thinking about that on a piece of paper. Or you can go in the other direction. You can look at a set of genes and maybe connect that back to a small molecule as a discovery tool. And this then obviously allows you then to also study biochemistry. So with this thought process that there are gene clusters, there are investigators out there look at this in public databases. This is from a paper by Michael Fischbach, who's at the University of California, San Francisco, and he looked at a number of publicly available genomes and realized there are tens of thousands, or at this time, of gene clusters, most of which we have zero idea the functions of the small molecules that arise from them. And so over the years, my lab has really been developing the tools and techniques to really connect genes, be able to read those genes, and think about them in the terms of being able to draw a chemical structure directly from genetic information like this. And this has allowed us to discover small molecules directly from genetic information. And over the years, we've worked a lot with people here at UC San Diego. Peter Dorstein, who's going to speak tomorrow in this session, is a remarkably talented mass spectrometrist, and he and I have combined forces in genome mining and from my group and mass spectrometry from his laboratory and developed new techniques to connect analytical chemistry directly to genomes to facilitate small molecule discovery. Actually, there was a workshop 2 days ago here on this campus, and of all places, over in the computer science department, who a number of investigators there are very interested in being able to think about this from a computer science point of view, being able to take genome information and predict chemical structures by these methods. We've used this approach to be able to connect chemistry to genes to do a number of things. A couple of years ago, I started a center at Scripps Oceanography for oceans and human health on a series of small molecules that are highly brominated, look like marine contaminant type molecules. They're produced by bacteria in the oceans. We now understand the genetic architecture of how they are made, what organisms make them, how they then move into the food chain and eventually get into humans where they have deleterious consequences. That story is for another day. What I would like to do with the remaining time today is I'd like to pose this question and then try to address this question. And this question relates to, we've done pretty well taking genes and be able to convert those into small molecules. But we would love to be able to predict a function of a small molecule from an orphan set of pathway genes before we even know the chemical structure of the molecule. So can we predict function based on just looking at a set of genes in a genome? So that's what I'd like to do. To be able to address that question, I want to take a step back and maybe just tell you a short story of how we came to this question and how we are now addressing that. And that relates to this marine bacterium that my colleagues at Scripps discovered about 20 years ago, now Bill Fennicle and Paul Jensen. And this bacterium, whose genome we sequenced 9 years ago now, dedicates about 10% of its genome to making small molecules. One of which is this small molecule, this very potent inhibitor of the proteasome, which is now in phase 2 human clinical trials as an oncology agent. And that molecule, the structure is shown there, and it sits in a sea of other small molecule pathways as well. I just want to focus on this small molecule, and I'll show you that on this slide, how this molecule works. I think nature's done a remarkable job designing this molecule. It's a pretty small compound. It has a lot of remarkable features associated with it, and if you begin to manipulate the molecule or change its structure, you often can lose activity. Nature's done a pretty good job with this structure. If you remove the halogen here, you lose 10-fold activity in vitro, about 1,000-fold in vivo activity. If you change this ring here to other things, you also lose activity as well. It interacts with the proteasome by being a suicide inhibitor, by interacting with the terminal threonine residue, which is the catalytic residue, to form this adduct here by opening up this beta-lactone ring. And then this halogen is then further cleaved in the second reaction to generate this adduct, which is now covalent and irreversibly bound. If you don't have a leaving group here, this type of species here can be hydrolytically cleaved off the proteasome. And that's why this molecule here has such less activity in vivo, about 1,000-fold. So there are two features of this molecule that are important from a design feature. One is this cyclohexene ring that fits into this binding pocket here. And then obviously this leaving group of the halogen here is also important. So from a bioengineering point of view, we want to understand how nature installed these units so we could begin to sort of think about whether we could diversify this molecule from a bioengineering point of view. So to do that, we sequenced the genome. We realized that there were a couple of natural molecules in the series, one with, one without a halogen. We did isotope experiments. We figured out this molecule comes from 3 different building blocks, an amino acid, an acetate, and 4 carbons on either side came from 2 different fates. Either a sugar pathway gave rise to these 4 carbons, or a lipid gave rise to those. Eventually, we figured out how nature assembles this molecule, and you know, I'm always blown away how nature can sort of synthesizes complex organic molecules and does so in a pretty, just a remarkably beautiful way of being able to take a number of genes whose, these 2 genes encode these 2 proteins here, which have 8 domains. 3 domains are these carrier proteins that carry 1, 2, and 3 different substrates. Each of these substrates, this red one and this blue one, all the arrows in red encode enzymes that synthesize this blue non-protein amino acid. All those in red make this one, stitch it together to make this molecule. If you think about this pathway as almost like our children think of as playing with Legos, if I put different pieces in each of here, I can maybe change profoundly the chemical structure here. If I can reprogram amino acids attaching here, I can change this unit here. That changes specificity. If I change this group here, I can also change reactivity. And that's exactly what we did. Through just biotechnology ways, we were able to generate a small library of molecules over a number of years by just reprogramming the organism to do this work. But that is also a departure from answering the other question. What we found at that time we were doing this work was in association with all the genes that encode the enzymes that make this molecule, there was one gene that just shouldn't have been there. And that gene encoded a proteasome subunit. So I was sort of curious because this molecule is a proteasome inhibitor, so why would you have a proteasome subunit in the cluster there as well? Well, you might think of this almost as an immunity factor. This organism, once it makes a pretty potent molecule, has to also protect its own proteasome. But you think, I didn't think bacteria made proteasomes. Well, actually, this bacteria and a few actinobacteria, which includes Mycobacterium tuberculosis, have the 20S proteasome like you and I. And elsewhere in the genome, there's a regular proteasome as well. So our thought process was, maybe when the molecule is being synthesized, it makes a resistant form of the proteasome. So we reconstituted the proteasome, the so-called resistant one, and the so-called sensitive one. We made those in E. coli. We tested it with this molecule, and we could see a 30-fold difference in sensitivity towards this agent. And looking at the sequence, we realized there was one residue that caught our eye, that was, in essence, conserved, whether it be in bacteria, whether it be in fungi, whether it be in mammals, was in this binding pocket, this residue is invariably an alanine. But in this resistant form, we saw that that was a little bit larger, and a valine was present there. If we do mutagenesis there, we see that this is one of the key residues for conferring this resistance, that this binding pocket has become a little bit smaller. So that's sort of curious. But even more curious, we found, was a study done around the same time by another group of people who took an FDA-approved proteasome inhibitor drug called bortezomib, used very high levels to induce resistance in human cells. And they were able to generate a mutant. And when they sequenced this mutant, they realized, in that case, they generated a mutant in which this residue had switched in mammalian cells from alanine to valine. So that's sort of cool too, right? Imagine you can think about sort of nature has, in this bacterium, evolved this form of resistance, but in humans we can induce this resistance form as well. So this got us thinking that, you know, let's say hypothetically we had no idea what the function of this gene cluster was as far as small molecules. But if I can recognize it makes a small molecule of some type, maybe if I didn't know what it was, maybe I should be thinking, since it's co-clustered with a proteasome subunit, maybe it would make a proteasome inhibitor. Maybe that's a way to do discovery. So we wanted to see if this could sort of hold some water. Are there other proteasome inhibitors out there? Yes, there are. There are FDA-approved molecule called carzilimib that just came out very recently, and it's an epoxy ketone molecule based on this epoxy-mycin molecule. We sequenced the genomes for the bacteria that make those molecules, and in both cases we found proteasome subunits associated with those gene clusters, and they had the same mutation as well. So that looked pretty interesting there too. So let's go on with that. So the linkage of immunity factors in biosynthesis is nothing new. That's been around for quite some time. We realize that's how resistance often gets transferred when we think about antibiotics. And in addition to the case I just told you here is that this oncology agent, this is a Merck compound called platensin, which is a fatty acid synthase inhibitor, and its target, which is the resistant form of FABF, is also present in the gene cluster. Here's a gyrase inhibitor called dovabiacin, and there's a resistant form of gyrase B there as well. This has been recognized in the field for quite some time. So we think maybe we can use this as a diagnostic to be able to do discovery. Because when we think about looking at gene sequences, and you have thousands upon thousands of genes that are out there, how do you focus on the ones you think that might be of importance? And we call this new technique target-directed genome mining. Now, to be able to play this game, there's one more little tidbit I wanted to tell you how to facilitate being able to interrogate function of an unknown gene cluster. What we'd ideally like to be able to do is take this set of genes from a bacterium, rapidly grab it, put into something I can move into another organism. And so we've been able to develop technology that sort of we borrowed from the cancer community and looking at something called transformation-associated recombination, which is a technique that allows you to transfer very large segments of DNA into a vector using yeast recombination. And a vector we designed that actually propagates in yeast, E. coli, and then a production bacterium. So we can transfer that DNA, we can move it into yeast, eventually we can deposit that into an E. coli organism, turn that pathway on, and cross our fingers we get a small molecule out of it as well. And we've done that, for instance, with this example here, where this is a pathway that's associated with a billion-dollar drug called daptomycin. We sequenced a marine bacterium recently that had a very similar gene cluster, but had some differences. It had differences in lipid biochemistry, one in amino acid biochemistry, and also in halogenation biochemistry there as well. We grew the organism up. It didn't produce. We saw that this pathway was transcriptionally inactive in the organism. So we thought this would be a great challenge. So we took that pathway using this yeast recombination technology. We're able to move that into yeast, move it into a host, transcriptionally activate the pathway, and generate a new small molecule that's here that was exactly as we predicted nature would make it based on the gene architecture, and this molecule has some profound activity as well. So now I think we're all set to do the question I asked at the earlier set of if we can sort of look at an orphan set of genes and associate it and be able to predict function before I know anything about its chemical structure. So we did this by answering or asking the question of if you imagine if I can find all the gene clusters out there associated with natural products, I can find those. There are tools, algorithmic tools that are out there that allow you to do that. And ask this question, the second question of, are there duplicated genes from the genome that co-cluster with those genes? And you can do sort of an analysis which allows you to do that. And this allowed us to sort of look at all duplicated genes from a genome that are coincident with a secondary metabolic pathway. And then you can associate this with any portion of biology, and we could look at sort of post-translational modification. We can find the cluster I just told you about earlier about salinospermide being present there because the proteasome subunit is present there. So if you have an interest in looking at lipid biochemistry or transcription or translation, you're just going to look at different categories and see which ones of those are highlighted with a certain gene cluster. So let's look at one example of lipid biochemistry over here. We found a number of lipid genes associated with small molecule biosynthesis. We found a number of genes encode FABF, which is a ketosynthase, which is part of making lipids. It turns out they fall into two families. One very large family associated with natural product pathways look like they just make lipids that get embedded into the natural product chemical structure. And we know that there are many natural products that have lipid components to them as well. We found one group of FABF enzymes that sort of coexisted with a resistant family associated with the molecule I told you about earlier, the Merck compound called platensin. But the genes that we found associated with that look nothing at all like that would encode for the molecule platensin. They look a little bit like this. So here's my gene cluster, here's my FABF that we predict is not a functional FABF in synthesis, but rather a resistant type. Here are those genes. They have nothing to do with the Merck molecule. We're able to capture those genes. We're able to then embed those genes in a surrogate organism. We're able to then turn those genes on to generate new small molecule chemistry. This is the chemical structures of molecules we got out of it. Turns out these molecules are FABF inhibitors. And they're quite potent inhibitors as well. They're called with these structures. And we were able to take this gene here and actually confirm that it was indeed a resistance factor by putting it into another bacterium and show that it conferred resistance. So we're taking this approach now in our lab to really interrogate pathways and be able to predict functions before we know anything about their chemical structure. It's an area that excites us right now. I think it gets us back to being able to do discovery, not from a grind and find way, but really using sort of hypothesis-driven methods to think about how we can sort of look at nature out there to generate new chemical entities. So I hope I've done with this talk to sort of share with you some of my thoughts on how discovery in the natural product field is a little bit different today than what it used to be. It's really taking the advantage of being genome science to be able to do discovery chemistry, being able to refactor or repurpose small molecules into different molecule structures, and also learn something about their mechanisms of action as well. So I'll end there. This is my group down at Scripps. You know, one of the best things about being here in San Diego is really having a number of colleagues, not just here at UC San Diego, which we have them down at the beach at Scripps or up the hill, Mesa here at the Med Center area, but also friends over at the Salk Institute, the Scripps Research Institute, and a number of others. This is a wonderful place to do science, and it's sunny most of the time. And usually I don't wear clothes like this. Usually I'm in shorts and a t-shirt. But for you, Palmer, I'm dressed up today. So thank you very much.
I think we have time for a couple of questions. So here, right in front.
This is really, really very interesting talk. I'm curious, do these gene clusters have a tendency to be able to undergo a horizontal transfer more frequently or less frequently?
More frequently. Yeah, absolutely. I mean, it's like bacteria have sex, right? They just transfer all these genes over.
But more than other clusters of genes, that's what I'm asking.
It's most like primary metabolism isn't, those genes in a microbe aren't clustered. They're scattered throughout and they're very differently organized. Usually secondary metabolism, you'll see this clustering. And maybe that makes sense because if you make a toxic principle, you have to have your immunity factor present with it as well. And so you wouldn't want those to not be coupled together.
But I think one of the points there was, are they being horizontally transferred more often than normal genes in the genome? And I think that the answer there is yes.
Yeah.
Quite a few of the compounds that you're showing, using as an example, Taxol, colchicine, quinine, these are all plant-derived.
That's right.
And I was wondering, is there an analogous system or that you can use for analyzing, searching for plant-related compounds?
Yeah, so super question. So, most of this work is taking place with microbes because, hey, they're a lot easier to work with. And, we've heard about, pick your system wisely, right, from the earlier talk. But plants have much larger genomes than a microbe do, so it's hard to get into it. But work that's being done now is showing that plants do actually pseudo-cluster their genes as well. They've just got much larger genomes, and the spacing between genes is significantly larger as well. But there is a pseudo-clustering in plants that's existing.
Well, I think with that we'll move along, but thank you, Brad.
Thank you.
Our next two speakers got together and decided to reverse the order, seeing that there was an opportunity to lay a foundation by doing so and make a nice series of presentations. So it is a change from your program. The next paper is given by Joel Sussman. Joel comes from the Department of Structural Biology at the Weizmann Institute of Science, and he's talking on acetylcholinesterase. 25 years since the 3D structure was solved. What have we learned?
Thank you very much. It's really a real great honor to be here. Can you hear okay? I must say, Palmer, I'm just struck by the students and postdocs that you have mentored. And this is one of the words, mentored, in the symposium. I think if one could find a way when you are hiring new young faculty, not to look at the H-index or the number of papers, but to try to predict the offspring. This, I think, is the judge of science. And I can just see from this room what is happening and outside of the room. So, I never had the opportunity really to work with Palmer, but I've known Palmer, I think, close to 25 years at various conferences. It's a small period compared to other people here. So I've collected a few photographs that I took of times. This was a meeting that was not a cholinergic meeting or cholinesterase meeting. It was a Eurotox meeting near Dubrovnik. And Palmer suggested at one point that we might take a day off. And we went to Dubrovnik on this incredibly small boat. I don't know if you remember that boat, but I wasn't sure either of us would make it both ways. This is Palmer getting instructions of where to go next. At the various 12 cholinesterase meetings that have been held around the world, Palmer has often played a very prominent role. Here he is with Elsa Reiner, giving an award, and there with the BP doctor, both of them unfortunately no longer with us. This was in China a few years ago. This is, for me, one of the most important events. There was a symposium similar to this for me a few years ago. Palmer and Susan came, and they met my father at the meeting. He often spoke about you, Palmer. I don't know if you know that after this, very frequently. And they came to my home, and this is my kitchen with Israel Silman drinking wine, of course. And finally, this was just a few months ago in Elke, where Palmer was introducing Rick as one of the featured lectures at the after-dinner meeting. So what I want to talk about is just briefly some work on what we've learned from cholinesterase, and not just about what we learned about cholinesterase, but it's a little bit like when we were talking last night with Jean-Pierre Changeux. He kept stressing that one shouldn't just look at techniques, but think of concepts. So I will get towards the end some new concept that came out, unrelated even to cholinesterase. This, I think, is very important. One of the problems we have in funding from Europe, I don't know if you have it here in the States, is you have to put milestones and deliverables into your proposals. And often when you do science, you begin with something and suddenly something you see, which is totally unexpected. And this I'll bring up something towards the end, which was just like that. So only thing here I want to stress from all the other part we've heard about is that the enzyme, the cholinesterase, is incredibly fast. It hydrolyzes 20,000 molecules per second, which is very important for the turnover, get rid of the excess acetylcholine so you can restart the receptor. It's also extremely specific for acetylcholine. Acetylcholinesterase doesn't hydrolyze even butyrcholine. So these two specificities are very interesting. There was lots known about the enzyme before the structure was determined, but basically, in essence, there was a steric site which was involved in binding the quaternary group, and a steric site which was responsible for hydrolyzing the ester, and the peripheral anionic site which was derived from work similar to early FRET, but it wasn't called FRET at the time, and it was thought to be about 18 to 20 angstroms remote from the anionic site, the second binding site. That was about as much as was known about the enzyme before the structure, except that the picture that's shown here is because the enzyme was so rapid, there was no doubt about one thing, that the catalytic site had to be on the surface. Otherwise, you couldn't have the substrate go in and come out. So this was always drawn right on the surface. So most everything was sort of right about this, except for this last point. So what I'll do is go over these points relatively quickly about where the anionic site, where the peripheral site is, because we'll learn about this more in Pascal's talk soon, about how it's so specific, how it's a member of a very large superfamily of alpha-beta hydrolase fold proteins. It has a very unusually high, large dipole moment, and what it might mean for some of the cousins, sequence cousins of acetylcholinesterase, and how acetylcholinesterase binds a number of cholinergic inhibitors, which are the basis of one set of drugs for palliative treatment of Alzheimer's. And then we'll get to something totally unexpected that came out from the work. So the work was done in very close collaboration with Israel Silman, really has focused, I think, most of his scientific career on acetylcholinesterase. This is one of his early students, Tony Futterman, and what they worked out early on was a way to purify the enzyme by using a phospholipase, specific phospholipase C, which hydrolyzed this bond here, separating a homodimer of the enzyme to get out the enzyme almost pure in a single step. And we used that to purify the enzyme very quickly. The enzyme was crystallized by Michal Harel, who's in the audience. She now spends 1 or 2 months a year here in San Diego working in Palmer's lab. So indirectly, we work very closely together this way through Michal. This is Michal, young Michal sitting here. She crystallized the enzyme. It was not an easy task, but she did it wonderfully, diffracted fine, and I won't go into how the structure was determined, but it was standard procedures now. And this was us looking at the structure on a computer graphics screen before everyone had a PC or Macintosh to look at things, so this was a major event to look at it on the screen. What we found, which was really unexpected, was that the active site, the histidine, serine, and glutamate at the bottom here, is at the bottom of a very deep gorge, about halfway into the enzyme, the opposite of what had been predicted. It isn't on the surface, and it's actually an extremely narrow gorge. This picture doesn't do justice to it. It's so narrow that, in fact, this region between the phenylalanine 330 and the tyrosine 121 is so tight that you can't even drag an acetylcholine down the structure. But it gets down there somehow. So this is the anionic site at the bottom. The peripheral anionic site has been now seen to be at the top of the gorge near this other tryptophan 279. And this is a cutaway picture showing the gorge going down like that from the top. This is an overall picture of the secondary structure of the enzyme. When we determined it, I sent it off to a former student of Adionat who was in Maryland at the time. I sent a fax. There was no emails. So we sent her a fax. I said, we really discovered something fascinating. It's a completely new fold. And then she wrote back to me that she has a student who just came from Jim Barrington's laboratory and had solved a carboxypeptidase 2 from wheat. And it was the same fold. I said, no, and it was the same fold. And there were 3 other enzymes that had this, enzymes that had totally no functional similarity and very little sequence similarity. The sequence similarity was under 20% of the amino acids in most cases, but they all had this fold, and it was a very large fold. Some of the sequences went up to over 500 amino acids long. And the position of the catalytic triad, which are shown in green balls, all lay at the same position in the sequence and in the same order. So this looks like a classic case of divergent evolution, where this fold was a good fold to hydrolyze something, and then diverged to different functions. There's now a database maintained by Arnaud Châtenay in France called the Ester database. This is the site, and there's over 45,000 genes that code for this kind of fold, which is really remarkably, so it's really one of the most popular folds, it turns out, and we thought we had seen something new in the world. When one looks at the binding of acetylcholine to the active site, this we published. It's a picture of the van der Waals surface of the enzyme. Then you can build in a model of how acetylcholine would lie with the quaternary group juxtaposed opposite the tryptophan 84 at the bottom of the gorge. This was a not well believed kind of idea at the time, because this is a quaternary group, it's positively charged. How could it be opposite a tryptophan? And there was lots of controversy, but later on we determined several structures of transition state analogs which show just this kind of picture. And in fact, there was a paper published from two scientists at Caltech just a few months before we determined this structure, showing a model of the acetylcholine receptor, which is a hypothetical model of the binding site, with a series of aromatic residues showing the quaternary group of acetylcholine sitting right in the middle. This is what they predicted, and this is what we saw in the enzyme. So we sort of began thinking maybe the enzyme and the receptor have similar structures even. This doesn't turn out to be true, but in fact, this picture of the binding pocket is somewhat similar in the two. We saw early on is that if one looks at the enzyme, with this being the gorge going down here, the northern hemisphere of the enzyme, colored red, is highly negative. There's huge excess of glutamates and aspartates in the northern hemisphere, and lysines and arginines in the southern hemisphere. So much so that it's got a very, very large dipole. If one calculates the dipole using Poisson-Boltzmann equations, one can get numbers close to almost 17,000 dBi for the monomer of acetylcholinesterase. And if one makes a simple histogram for the number of proteins that have this size dipole, one can see that acetylcholinesterase is one of the largest dipoles. So this, we thought, must be related to the fact that the negative charge would pull down the acetylcholine into the gorge. It may be related to that, but there's a lot of evidence now that maybe it's not true. I won't go into all the details now why. But when we look at the structure, we could picture positive charges going in, down. But as I said, it's very narrow here. The enzyme hydrolyzes acetylcholine down here, and there's really no way for it to go back up the gorge without having a bottleneck, sort of like in Los Angeles all the time with the cars. So we predicted, based on some simple modeling, that if this tryptophan 84 would just slightly change its conformation, it would open up a back door, and the substrate could come in, and then the product could be released. This was also very controversial when we first proposed it. I think we proposed it in the meeting in Chennai, in Madras. And the reason it looked controversial is because it looks like it defies all the laws of thermodynamics that goes one way. But now there's a lot of evidence, which I won't go into now, but based on papers that we've written with colleagues in Grenoble, from Martin Vecht's laboratory, doing studies of putting in intermediates and trapping them to actually see the opening of this back door. But the big dipole may be related to something totally different in the enzyme. One looks at fasciculin, which we've heard quite a bit about in the meeting. Fasciculin comes from the venom of this pleasant snake, the green mamba, and it has a lot of terrible things in the venom, but one of the components is a 61 amino acid peptide, fasciculin, and it also has a dipole, the structure, but it's only about 100 dBi. So if one pictured the structure of fasciculin and cholinesterase, the dipoles could line up, and then we determined the structure, and Pascal and Yves and Palmer determined the structure, all at the same time. We were talking to each other, actually at that time, and working at the same synchrotrons even on it. Palmer and Pascal and Eve determined the structure of the mouse acetylcholinesterase with fasciculum. We determined it with torpedo. But what one sees is that, in fact, it comes down like that, and the positive charge is attracted to the negative charge. It gets together. What's interesting is that all of the moles had the positive ends, the arginines, the lysines sticking out and making wonderful interactions with the carboxylates of the negatively charged groups. But when one looks at the structure in detail, in fact, the reason why fasciculum binds so tightly to the enzyme is it doesn't bind like that with the side chains, but the side chains interdigitate. The positive ends of the arginines and lysines go and bind to the carboxyl of the main chain of the polypeptide chain, and vice versa. So they interdigitate, and the enzyme doesn't come off. The toxin doesn't come off from the enzyme. So this is a picture taken from the paper of Eve, Pascal, and Palmer, showing the mouse enzyme in green and the fasciculum shown in red, with these fingers sticking in and forming these really tight interactions. What one sees, and I don't think it's been stressed enough, is that there may be close to 100, maybe over 100 acetylcholinesterase structures solved by various groups now from many different organisms, from Drosophila, human, torpedo, mouse. In every case, even if the enzyme was purified apparently as a monomer in terms of the way it was cloned, it always crystallized with this very clear twofold 4-helix bundle formed from one monomer and then the second monomer. This always shows up that way. One looks at this fasciculum bound to the enzyme, it almost looks a bit like an adhesion protein interacting with it, the way it's sticking on. And in fact, this slide I received from Palmer, I think in the early 1990s. He showed this, and I asked him permission to get a copy. This was a picture before all the structures were known, but it's a phylogenetic relationship of a number of proteins, including 3 of them here: neurotactin, gliotactin, and neuroligin, which aren't in any way enzymes. These are, in fact, adhesion proteins, and we'll hear a lot more about this, I hope, from Pascal's talk. No? Paul. It's okay. Thank you. All right. So these 3 are here. The only difference is they don't have all of the members of the catalytic triad. They don't hydrolyze acetylcholine. And they are adhesion proteins. And this is a picture taken from a paper from Palmer, Eve, and Pascal, and Fabergé, showing the interaction in this case of neuroligin with acetylcholinesterase. And it just looks just like the way fasciculum binds, almost. Because when you look at it, in fact, this is when you look at it, this is where the neuroligins bind, just the opposite side. And so it's clearly not the same. It's different from what everyone also predicted. Everyone assumed it would be up here. So we'll hear more about that later on. I don't want to steal thunder. Cholinesterases have been used now as the target for a number of drugs that have been developed for treating in a palliative manner the early onset of Alzheimer's. One of the most widely used drugs is Aricept, and in our lab we've studied a series of these drugs and how they actually interact in an atomic way, molecularly, with the enzyme. It's been seen that in model studies that the level of acetylcholine is reduced in patients with Alzheimer's. So one of the approaches, which has been thought for a long time, that might alleviate some of the symptoms of the disease is if you could reversibly inhibit the acetylcholinesterase enzyme, you would then operationally somewhat raise the level of acetylcholine and restore some of the cognitive functions. This is the basis of this class of inhibitors. So we determined the structure of all these molecules. On the right side, the molecules are synthetic ones. On the left side, these are natural products. mostly from moss and from plants. What's interesting is how different they look chemically from acetylcholine, but all of them bind at the active site. Some of them bind longer. For example, Aricept binds from the bottom of the gorge all the way to the top of the gorge. And this is a picture of showing in green the Aricept molecule bound from the bottom of the tryptophan 84, going all the way up to the top to tryptophan 279, with a number of water molecules lining the surface of the gorge. And what we found, which was really, to us, quite amazing, when one looks at the structure of the acetylcholinesterase without the inhibitors, there's a series of about 18 to 20 water molecules in the gorge. When you put this very large inhibitor in, only 3 or 4 of those water molecules are expelled. The others stay in the gorge, and in some sense, they actually are helping to determine the specificity of the inhibitor bound in the gorge. This molecule and the other cholinesterase inhibitors, as contrasted to what Gabi was talking about before, are all reversible inhibitors. For the obvious reason, if they were not reversible, we might have some of this effect. On the other hand, I think if someone took one of these medicines, or an elderly person took the medicine, and the results look good, the obvious thing to do would be to think, well, let's just double the dose and double the dose. And in some sense, if you did that, it would probably result in something like what Gabi was describing with the irreversible inhibitors. So these are reversible. They don't bind in any way except with aromatic pi-pi, cation pi, aromatic H-bonds, and binding through water molecules. So there's almost no direct interaction with the protein. So now I'll just get to a few of the surprising results, and then we can have a coffee. So the first is a little bit about the flexibility of acetylcholinesterase. The enzyme is in some ways surprising. It's really not a very flexible enzyme. There's no loop that's moving back and forth, as we heard earlier, the morning from Scott's talk. If one looks at the structure and lines up many of them, there's small changes here and there. I think some of the small changes are crucial, especially some of the changes in side chain. But so far, no one has observed massive changes in the structure. So we'll talk about that. And then the second part is some of the work we did to try to visualize the enzyme's actual activity and motion. So this work on the flexibility was done by a wonderful postdoc, Yichun Zhu from Shanghai, and Harry Greenblatt from our lab in the Weizmann. She came and worked 4 years in our lab. And the work was done on a natural product, a natural traditional Chinese medicine called huperzine A that comes from a small moss called Huperzia serrata in southern China. It has been traditionally used as a leaf from the moss, put into hot tea, you let it brew, and then you drink it, and you have much better memory in the literature. But it was studied in China, in Shanghai, and in Hong Kong together after the Cultural Revolution. And the leaf was examined, broken down, and the chemicals in the leaf, a little bit like we heard before, but using more traditional methods, to try to see if there was any activity. And one of the activities that was seen by Professor Tong was that it is a potent inhibitor of acetylcholinesterase. And so this is why the Chinese began studying it as the purified compound from the leaf. Yichun did a lot of this work for her PhD in China, and then came to the lab to study molecular biology. But she took some of her work, which was doing a molecular dynamics simulation for her PhD, of seeing how huperzine could go down this gorge because it was too fat to get through this region. And I'll show you what I mean. I have a picture of how huperzine would go in if you didn't have molecular dynamics. This is for you, Andy. All right? And so this is the structure of the enzyme, showing a space filling with the residues lining the gorge here. This is the active site serine right in the middle. And the huperzine will come along and just crash into the structure. And I think this picture is in some sense one of the things as a structural biologist, as a crystallographer, I can say where we've done a misnomer. We've done a bad deed for the world. I think in general, protein structures are not rigid. And one has to always remember that one looks at, this is the case where we see the actual crystal structure, we determine this in our lab. It fits beautifully in the bottom of the gorge. And I will skip further on because I don't want to waste the time. Now, what Yichun did, she saw that it couldn't go that way, so she did a very simple study which is called steered molecular dynamics, where she takes the huperzine and she gives it a small force in addition to all the electrostatic and conformational forces and van der Waals forces, but she gives one extra force, which is a force to slowly pull it down the gorge. And each stage as she pulls it down, she lets the molecule relax and does a computational equilibrium, computation to determine the structure at each stage. So as she's pulling it down, she gets the structure and she sees how the structure changes to allow it to get to the bottom of the gorge. It's clear this is only a theoretical study, but what's important is you can see how much the protein has to change, almost as part of the protein is pushing it down. But at this point, this region where the tyrosine and phenylalanine are open up so that the huperzine can get to the bottom of the gorge. And it's clear without that, it would never get to the bottom. And I think this picture, to me, I think helps explain why proteins are so large. Proteins are large so that you can have an active site at the point, but have enough flexibility to get to the active site as well. So this last part, which I'll just go through briefly, but I think it may be of interest also in the same sense of what getting at before, of other problems which I think crystallographers sometimes sweep under the rug. So listen carefully. This is an attempt to try to visualize the catalytic reaction. And what we wanted to do was to put in a cage compound into the enzyme and then flash it with a laser very rapidly and then collect the X-ray data in microseconds or even faster. And so this was the goal of the experiment. But what came out was, from the control experiment, something even easier, something even more interesting. So the work was done in collaboration with Raymond Ravelli and Martin Weck, two young, extremely good scientists whom we met in Grenoble. Raymond was working at the synchrotron. Martin was working at the Institute for Biological Structure in Grenoble. And this was the goal, to really do stop-action video, almost like going into a discotheque and shining the light as you watch the people dancing. It resulted in a very interesting paper, which we'll come back to afterwards, where we actually could watch a chemical bond, but not the bond we wanted to watch. So I suggested before we start, we should maybe do a simple control. And the control would be to take a protein crystal, one crystal, irradiate it rapidly, collect the data, stop, and then do it again, stop, and so on, to make sure there was no artifact in collecting data very rapidly. I think I had criticism, but they agreed to do the experiment. And so we did this experiment at cryogenic temperatures, so this was not yet the experiment to see the action of the enzyme, just to see what the radiation did. And the general theory, so the radiation may, even cold temperatures, might cause a little bit of damage, but nothing very interesting. And what came out was something totally unexpected. We saw that when we irradiated the crystal this way, the same crystal, up to 9 times, we saw a disulfide bond actually break. We could watch the breaking of the bond. We could see a carboxylate, the carboxyl acid of glutamic 327, the active site, carboxylate, actually disappear. And we could see some increased disorder in the active site. I won't go into all this, but the beam is extremely strong on a synchrotron. It's 1,000 to 10,000 times stronger than a local source, so it's really something strong. But the common wisdom, until this work was done, was that if you radiate a crystal at cryogenic temperature, really nothing happens. This is what the cryogenic temperature is protecting the protein from. So we did 9 successive X-ray data collections on one crystal. We waited 15 minutes between each data collection, and then started again. And initially, the first set had 2 action resolution. By the 9th set, it was only three axioms. There was overall some decay in the protein. Without the details, but the overall quality of the structures were good. Each structure was refined independently from the starting model. In fact, what happened is we collected the data in Grenoble. I wasn't there. Raymond and Martin were there. They flew to Israel. At that time, we had very good computers. And over the weekend, we decided to refine the structure. And we thought this would be the preliminary, and then they would go back and do the real experiment. And this is not what happened. So they did the calculation, and then we saw the results. So, analyze the results. The SS bonds were not included in the refinement. We didn't put the disulfides in, so we could just let them come out, almost like the way you'd see a ligand coming out when you do putting a ligand to see where it is. We wanted to see the disulfides and so on. And this is a picture of one of the disulfide bonds. Over here, it's a 3FO minus 2FC map, so it shows all of the atoms. This is just a difference map, so you'll see the disulfide. So initially, you can see from the first data collection, a beautiful disulfide bond. The S and S would be over here. Already by the second data collection, it begins to break. And by the 9th one, there's no disulfide bond left at all. So the X-rays basically destroyed part of the protein. We looked at a second disulfide bond. Same way, and nothing happened to it. It survives just fine. So the radiation damage is very specific. So if you were looking at this and you only collected data in a normal way, and you went back to the previous slide, this final picture where there's nothing left, if you had collected the data in a conventional way, you would have had equivalent of 9 radiations. You would say that that disulfide bond was broken. So in some sense, the actual experiment of the X-rays was changing the picture of what you were seeing. If we go further, this is a picture of where this bond is. This is the one that's broken. This is the one that wasn't broken. And I didn't show you this one, but that one was also not broken. So the broken bond was on the surface. So I'm not sure it's always the case that it'll be the surface bond, but in the second, this was the torpedo structure. In the human structure, that same bond we saw was broken when we collected on the normal way. So I'm convinced that that bond was probably broken by the X-rays. This is a picture of the glutamate 327 over here as a function of the 9 exposures. By the 9th one, almost all the electron density is gone. So you can actually watch the electron density disappearing for this. And this is the case for many of the glutamic acids in the protein structure. So the conclusion is that SS bonds are highly susceptible. Possibly the deeper they are in the protein, the less sensitive they are. Carboxylate residues, I didn't show them all, but many of them lose their density. But the dose for this very strong dose is very specific. Certain parts of the protein are affected, and other parts are not. It's not uniform. And you can read about it in this paper. So the take-home lesson is that faster and stronger is not always better. Don't always seek the biggest and brightest beam. You might be altering your sample in not a clear way. And in some sense, although we were chicken when we sent the paper off for publication, we felt it was a bit like Heisenberg uncertainty principle, where Heisenberg uncertainty principle states that it's impossible to know both the precise location and the precise velocity of a particle, since the act of observing one changes the other in a definable way. So in some sense, this now is well known, and it's a big problem in data collection for macromagnetic crystallography. And many people are trying to work on ways to overcome it. When we tried to publish the paper, we couldn't get it published. We sent it to PNAS, and they said, no, it's not true. If it's cryogenic, and no synchrotron in the world would be honest to say that they put a huge piece of lead in the way of the beam to attenuate the beam before the X-rays hit the crystal. Finally, we got one colleague in Argonne to put it in personal communications, and then the editor accepted it, and they put it on the front page of PNAS after that. But it was a case that they built these strong synchrotrons, and they didn't want to say that they were causing trouble for the crystals. So in closing, what we found was the nature of the anionic site, the peripheral site. We saw the specificity of how tight the pocket was with acetylcholine. The large dipole appeared to be involved much more in the binding of other molecules, such as the fasciculum, not necessarily related to the way in which the traffic of the substrate related. The functions of the cousin enzymes were later on derived by other groups in ourselves, but this was a spin-off. We saw limited flexibility of the enzyme, but it's clear without that limited flexibility, the enzyme wouldn't function. And the X-radiation damage, very specific rather than a random phenomena, was a real big surprise and spin-off from this work. The work was done by a lot of people in our lab, but I want to give particular thanks to Lily Toker, who's not here, but she purified hundreds of torpedo samples until we began to clone and express it ourselves in the human form and others. And Michal Harel, who is here, who was crucial throughout all of this work, in crystallizing and turning many of the structures. And it's been supported by Dietra and by the Weizmann. And in closing, I want a picture of two very important people, and to congratulate Palmer. This was taken also at the symposium where you guys came, and I really thank you very, very much.
I think given the time, we'll just take one question. Yeah. For coffee break? Yes, Andrew.
Just a quick question. You're suggesting, I gather, that in the radiation damage, bonds are being broken. Is it possible that there could be conformational isomerization and local structural disorder?
Oh, I'm sure there could be that. And not only that, Andy, people have used, Raymond Ravelli has worked out a way to use the radiation damage as a way to get phasing. To collect the data before and after radiation damage, it's as if you're taking out a heavy atom from the structure. You're taking out a sulfur. And it's been used to phase structures in a sense. So I'm sure there's also some small changes that could happen. People have seen changes also in unit cells.
Okay, with that, let's thank our speaker and